All right, our last lecture in the set for this week is going to look at environmental sample processing, and we're specifically going to look at soil. So this is chapter 8.1 in your textbook, and the next couple labs that we have are soil labs. We're going to talk a little bit about how samples get collected and a little bit about how they get processed. So we know soils are very, very highly variable environments, and we're going to learn a lot about the soil environment in the coming weeks. But many times the most accurate approach, because of this high variability, is to take many samples within a given site and to do separate analyses of each sample. And that's because there's a lot of variability. So you could move a meter away from where you initially took a sample, and the characteristics of the microorganisms in that soil are going to be very, very different. But many times what we can do is we can try to minimize the amount of material and the time that we use by combining samples taken to form a composite sample. So many times what we do is we have different ways of sampling a site that we'll go through, and we will find a way to create a composite sample of all of those as a way to broadly characterize what's going on in the site. And that's going to help us to limit the number of analyses that need to be performed. Sample analysis in the microbial world is very, very time intensive and material intensive. So when we can find ways that are going to give us at least a sort of a modicum of accuracy that would help us, we try and do that. So one way to do that is to come up with what's called a quality assurance project plan, or a QAP. And this is something that you may see if you become somebody who does a lot of sampling in the field, but is just a delineated sampling strategy that ensures the quality of the samples is not compromised. So basically, a QAP from your book is just a way of making sure that we have a rigorously documented sampling strategy, not only from the method standpoint, but from the storage standpoint and how we're going to collect it, so that we have that kind of plan for how we're going to do it. So we, as your teachers and your TAs for this class, we choose the types of samples from different plots for soil, and we think about what type of soil do we want to test, how many samples are we going to take, how deep do we dig, those kinds of things. So we don't have an official QAP for how we collect soil samples for our labs, but in the field, if you are working on collecting soil samples, this is something you'd want to have. So a lot of times it's very easy to obtain soil samples with just using a shovel, or what we call a soil auger. So that is what the picture is on the bottom of the slide. A simple hand auger is something that takes soil samples from very shallow areas that are in the unsaturated layers. The augers are going to be more precise than a shovel, obviously, if possible, because it's going to get a very thin tube of soil at various depths. And it's used for that incremental sampling depth. So we can go anywhere between 30 centimeter increments to depths and increments of 180 centimeters. It depends on the tool. So the benefit of that is that you go straight down and you can do increments of specific depths. A shovel is a little bit more of a simple way of doing things. It's not bad, and we use shovels for this class. But if you want to get more precise, you need more precise instrumentation. Most of the time we are going to be sampling what we call surface soils in this class. We don't go very deep to the subsurface down towards the bedrock. And we'll talk about all these soil layers as we get more into the soil environment in class. But many times what we need to do when we're sampling surface soils is we need to have a strategy to understand what we call 2D spatial orientation variations. So all the pictures on the right are going to show you different ways of thinking about how to take a sample. So this one right here, A, is called a simple random, where you take random samples. B is called a transect, where you find a specific line and go across that line to see variations along a specific line. You can do two-stage sampling, where you make a grid and you take two samples within each square of the grid, or you can do a systematic grid, where you take a sample within each of those. And they're used for different areas, or for different reasons, right? So if you were researching, or if you were taking soil samples and you wanted a very specific result, you would choose one of these types of spatial orientations. And random is actually not a bad thing. Sometimes we think of random as maybe being bad, but sometimes that's actually not that bad for specific types of things. If we were trying to look at a very, very specific set of variables, like say, for instance, we wanted to know how fertilizer application impacted soils, we'd want to be much more systematic. So a systematic grid or a transect grid or duplicates using the two-stage might be more appropriate. So just remember that it's costly and time consuming to sample everywhere, so we do need to have some kind of a strategy as we sample for soils. So this slide just explains those different types of sampling, right? So the random is just choosing those random points. The transect is specifically along one specific area. The two-stage has those regular subunits and has duplicates, and the grid sample is systematic. So feel free to take a pause on this slide to write down these in your own words. That might help you remember what they are. And these are just some different ways of thinking about how we want to take our samples. So one thing that I want you to give a try in these little pre-lecture slides is we have a scenario here and we want to design some kind of a soil sampling program. So we have a site that's being evaluated for suitability for land application of waste. The area chosen has been used for agriculture in the past, and application of waste is being considered as a way to maybe give us more nutrients for our crops, which is great. But we really don't know a lot about what the indigenous microbial communities in the soil are. So take a moment and think about this. Take a pause in the video. What kind of sampling would you recommend and why? So if we're doing these 2D types of sampling, right, remember that this is just on the surface, right? What does this not tell us? Well, it doesn't tell us variation with depth. So variation with depth is also very important. So sampling soils is kind of difficult because we really do have a 3D space that we're dealing with. So the 2D sampling gives us an idea of variation spatially on a plot with like an X and a Y, but the depth is actually very important when it comes to soil as well. We're going to talk about all the layers of soil in class, and those different layers are going to have different types of organic matter, different types of microbes, different indigenous components. They're going to have different minerals, all that types of stuff. So that's one thing to just be aware of when you design some kind of a surface sampling plan, is that you're not quantifying depth. So let's talk about depth, right? It can be very, very complicated, right? It's very challenging to deal with specific areas with bacteria and roots and things like that. But actually taking samples at increments is not that hard. It's just that it's sometimes difficult because there's a lot of changes with depth. So we could take things at 50 centimeter increments, that's kind of standard in the industry, down to a depth of 200 centimeters. Or we could have this very complex drilling several hundred meters down into the subsurface, into like the vadose zone, which we'll talk about in class. So you could take very, very small amounts of samples down in just the surface, or we could get way down into the subsurface. So if you're somebody who's interested in geotechnical engineering and foundations and how that interacts with environmental engineering, you'll get involved with some of this types of soil samplings. And the other thing that makes it difficult when we do soil sampling for surface soils is that we have plants and we have life in surface soils. And so one thing that's a little bit difficult is that we often end up sampling or interacting with the rhizosphere. So the rhizosphere is that specialized zone of soil that's under the influence of plant roots. Many times there's symbiotic relationships between microorganisms and the plant roots. So there's normally a lot going on. So when we take samples for the class for you, we have to take it out in a field where there's a lot of grasses and there's a lot of root structures for the grasses. And we have to be very careful to make sure that we move around those plants and dig just deep enough or away from roots if possible, so that we're not getting the influence of plants, because plant cells are going to be different than bacterial cells in soil. And so that's something that can be a very big confounding factor when we talk about what's going on in the soil. So if you're sampling in the rhizosphere, there's a couple things to think about. The rhizosphere soil basically exists in what we call a continuum with the root structure. to the point where the root has no influence. So it's like this whole area. So it's generally only about 2 to 10 millimeters, but those soil volumes are variable and they're difficult to sample. So normally what we do is we carefully excavate the roots and we shake very gently to try and remove the bulk or the non-rhizosphere soil. And then the soil that adheres to the plant roots is considered to be the rhizosphere. It's not super, super technical in terms of how we do that, but it is actually pretty accurate. And then if we sample the rhizosphere, right, what's going on in the rhizosphere is kind of difficult to categorize. So it still remains kind of a major limitation when we're looking at what's going on for microbial analyses. We're not going to get the things that are interacting with the plants. And that may be a good thing, but it also may be a big challenge if we're looking for interactions within an environment. And then lastly, below the rhizosphere, below the surface is what we call the subsurface layer. So these are the layers that are deep in the soil. They're going to have less microbial activity, but rotary drilling is going to be the most common in order to sample. So we need a drill that basically spins. It's like a coring device and it goes down. And it's used when we're trying to get into those lower layers or when we're worried about where the level of saturation of water is in the soil. So if we're in the unsaturated area where there's not a ton of water and there's still air spaces in the soil, we use what's called air rotary drilling. But if we get down into the saturated area where there's lots of water and lots of potential for soils to move on us, we use what's called hollow stem agar drilling with what we call a push tube sampling. So it looks just like the traditional hand agar tool, but it's in a drill. And it's specifically set up so that the water is having minimal impacts on the soil sample that we're pulling up. So that's something to think about. So those are all of our different types of sampling and thinking about getting soil from the environment. Once we have the soil, then we need to think about performing those analyses as soon as possible. One of the challenges with a soil sample is the moisture content related with the soil. And moisture content is huge when it comes to soil. If you've changed it a little bit, you've changed the microbial community completely. So moisture is kind of a big thing when we think about soil sample processing. So most of the time, the steps when it comes to soil sample processing is to first air dry the sample to remove a lot of that extra moisture, because a lot of that extra moisture is going to influence the weight of the sample and what's going on in the sample. And then normally what we would do is we would sieve the sample through mesh to try and remove a lot of big particles or rocks or roots or things like that, so that we're just getting under the soil level. If we're going to store it short term, we store it at 4 degrees Celsius, and we have a lower temperature here because we want to make sure that we're not getting a ton of growth, because a lot grows in soil because there's so much there. And then we can actually store those samples for about 21 days without losing data about microbial communities, which is really nice. So we actually will store your soil samples for quite a while. And one thing that's interesting about soil sampling is that we actually don't require super, super sterile conditions. And that's because, again, soil has such a huge microbial mass associated with it. So that's really critical. Last thing that we'll talk about is why should we take care with sample processing? So a couple things. Soil samples have lower culturable counts. So compared to water and air, it's harder to culture from soil, which means that sometimes other microbial contaminants could, or other microbial communities could affect the numbers that are being counted. So they're less likely to be culturable, which is important to think about. Then the subsurface stuff that's way down underneath the surface, they're not routinely exposed to the atmosphere. So it's very easy to have contamination from the atmosphere, microbes floating around in the air, contaminate those soil and contribute to a high volume of microbial types found. So making sure that those samples don't come into contact with a whole lot of air before they're processed, or processing in a fume hood or things like that, is going to be important for the subsurface specifically, because they're normally not exposed to the atmosphere. And then lastly, it's more expensive to obtain those subsurface samples. So many times there's no second chance at collection. So you really do have to think about making sure that you've collected in the right locations. So that's going to be soil sample collection and processing, and then we're going to talk more about the soil environment as we start to move forward in the class.